Before forests covered the land, before animals moved through water or air, before the sky itself turned blue, life on Earth began small, almost impossibly small. There were no eyes to see, no brains to think, no voices to speak. There was no intention, no awareness, no sense of self. And yet, these first living cells would change the planet more profoundly than any organism that came after them. To understand life as we know it, we must return more than 3.8 billion years into the past, to a world still young, unstable, and hostile, and witness the rise of the first true inhabitants of Earth, the Prokaryotes. By this time, Earth was no longer entirely molten, but it was still far from gentle. The planet had begun to settle, but only in relative terms. Volcanoes erupted frequently across the surface, releasing immense quantities of heat, ash, and gas into the atmosphere. The air remained thick with carbon dioxide, methane, and other volcanic gases. Oxygen was still almost entirely absent. Without an ozone layer, ultraviolet radiation from the sun reached the surface unfiltered. The sky was dim, hazy, and chemically aggressive. The oceans covered much of the planet's surface. They were warm, restless, and saturated with dissolved minerals. Iron, sulfur, and other reactive elements circulated through the water, driven by tides, geothermal heat, and constant volcanic activity. These seas were not tranquil. They were dynamic chemical systems, constantly stirred and reshaped. From a distance, Earth still appeared barren and lifeless, but beneath the surface of these ancient waters, something irreversible was underway. Chemistry had crossed a threshold. Matter was no longer merely reacting, it was beginning to organize. In shallow coastal regions, near volcanic islands, and along the flanks of hydrothermal systems, complex chemical networks persisted longer than ever before. Fatty molecules assembled spontaneously into membranes, forming thin, flexible barriers. These membranes were fragile, easily disrupted by heat or turbulence, but they represented a profound shift. For the first time, chemistry could be enclosed, and inside could be separated from an outside. Reactions could occur within a protected space. Inside these primitive membranes, chemical processes became localized. Molecules interacted repeatedly rather than dispersing into the surrounding ocean. Energy could be captured and used instead of dissipated. These systems were not yet fully alive, but they were no longer merely chemical accidents. They marked the emergence of the first true cells, prokaryotic cells, lacking nuclei and internal compartments, but capable of maintaining themselves against an increasingly hostile world. These early cells were simple by any modern standard. They contained no mitochondria, no chloroplasts, no complex internal structures. Yet their simplicity was their strength. With minimal requirements, they could survive where more complex systems would fail. They fed on chemical energy drawn directly from their environment. Hydrogen, sulfur compounds, iron, and other reduced molecules served as fuel. These cells did not rely on sunlight, they relied on chemistry, exploiting energy gradients wherever they appeared. Within their membranes, genetic material emerged in primitive forms, short sequences capable of being copied, though not perfectly. Each time a cell divided, mistakes occurred, errors accumulated, variations appeared. For the first time in Earth's history, information could persist across generations. Natural selection began its work, not as a dramatic event, but as a quiet filter. Cells that copied themselves more reliably survived longer. Cells that harvested energy more efficiently divided more often. Change became cumulative. Survival, however, was never guaranteed. The world these cells inhabited would be lethal to most life today. Ultraviolet radiation bombarded the surface waters, damaging molecular structures. Meteor impacts, though less frequent than before, still occurred regularly, reshaping environments without warning. Ocean temperatures fluctuated dramatically, sometimes approaching boiling in volcanic regions, then cooling as activity waned. Chemical conditions shifted constantly, altering pH and mineral availability. And yet, prokaryotes endured. They adapted rapidly, exploring every possible ecological niche. Some thrived in the depths near hydrothermal vents, feeding on hydrogen and sulfur released from the planet's interior. Others colonized shallow waters, developing mechanisms to tolerate intense radiation. Some learned to metabolize iron, using it as an energy source. Others exploited methane or carbon dioxide. Life diversified not through comfort, but through relentless challenge. Each new environment forced adaptation. Each adaptation increased resilience. Over hundreds of millions of years, these microorganisms spread across the planet. They colonized oceans, sediments, and mineral surfaces. Wherever liquid water and chemical energy existed, life followed. In shallow seas, dense communities formed. Layers of microorganisms grew atop one another, trapping sediments and minerals. Over time, these layered communities hardened into structures known as stromatolites. These formations were among the first large-scale biological structures on Earth, built slowly, layer by layer, by countless generations of microscopic life. From orbit, Earth would still appear barren. There were no forests to reflect green light, no animals to move across the surface. But at the microscopic level, the planet was alive. Every drop of water teemed with activity. Chemical cycles driven by life began to rival those driven by geology. Life had become persistent, global, nearly unstoppable. Inside these tiny cells, a profound transformation was underway. Metabolic pathways became more refined. Chemical reactions that once occurred sporadically were now regulated. Replication became more accurate, though never perfect. Repair mechanisms emerged, allowing cells to survive damage that once would have been fatal. These processes unfolded slowly, over immense spans of time, invisible to the naked eye, but foundational to everything that would follow. Some prokaryotes would eventually discover a new energy source, sunlight. Through simple forms of photosynthesis, they learned to capture light and convert it into chemical energy. This innovation did not immediately transform the planet. At first, its effects were subtle. But over hundreds of millions of years, photosynthetic life began releasing oxygen as a waste product. Oxygen accumulated slowly in the oceans and atmosphere, altering the chemistry of the planet itself. For most early life, oxygen was deadly. It damaged molecular structures and disrupted metabolism. Entire communities perished as oxygen levels rose. But some cells adapted, developing ways to tolerate or even exploit this new chemical environment. Life did not retreat, it transformed. Other prokaryotes formed intimate partnerships. Some cells began living inside others, exchanging resources and protection. These early symbiotic relationships laid the groundwork for one of the most important transitions in the history of life, the emergence of complex cells. But that revolution lay far in the future. For now, life remained microbial, small and patient. For nearly 2 billion years, prokaryotes ruled the Earth. They shaped the oceans, altered the atmosphere, and restructured the planet's chemistry. Every ecological system that would later support plants and animals was prepared by these ancient microbes. Every tree, every animal, every human cell traces its ancestry back to these early forms of life. Life did not begin with grandeur, it began with persistence. In a world of fire, poison, and radiation, the smallest forms learned how to survive. And in doing so, they reshaped the planet itself. We are not separate from this story. We are its continuation, carriers of a lineage that began in warm oceans, inside fragile membranes, guided by chemistry and time. To understand the first cells is to understand ourselves, not as masters of Earth, but as one of its oldest experiments, still unfolding.